Okay, this lecture is going to be on the reproductive system. And the reproductive system is a very effective system of the human body. And so we'll start ourselves off with a little bit of comics here. You can go ahead and take a moment to read that. All right, so the reproductive system in humans is very well designed. It's extremely efficient and incredibly effective. It is designed to reproduce and will find a way to do so despite some contraceptive efforts. The reproductive structures begin to develop in fetal development, and there are homologous structures between male and female humans. So homologous means grown from the same fetal origin tissue. And so in figure 17.3, we can see that they've color-coded some of the regions of tissue at about 6 weeks into fetal development. So this isn't 6 weeks after birth. This is 6 weeks as a fetus. And you can see that the yellow portion of the tissue in the fetus is going to either become the glans penis in the male or the clitoris or glans clitoris of the female. So they are structures of the reproductive system between the male and the female that were grown from the same fetal tissue. We can also see that the tissue in blue is going to become the scrotum and prepuce of the penis in the male, and it would become the labia majora, so the outer folds of the vulva in the female. And then we have the pink tissue, and the pink tissue from the fetus is going to become the urethral fold and ultimately a structure called the perineal raphe in the male along the penis, and in the female it becomes the labia minora, or the more medial folds of the vulva. So all of these structures grow from the same fetal tissue. It's just whether or not the fetus produces testosterone will determine what that tissue will become. In the male fetus, testosterone is present, and therefore that tissue will produce male structures. In the female fetus, there is no testosterone, and so it will produce female structures. Now, the internal duct structures of the reproductive systems. So in fetal development, at one point, 2 types of duct systems are present, and they are the Müllerian ducts and the Wolffian ducts. Now, in fetal development, one of the 2 ducts will recede, meaning atrophy and be resorbed by the body, and the opposite will do the same thing for the other gender. For males, the Müllerian ducts recede, so they go away. And in females, the Müllerian ducts become the ducts and glands of the female reproductive system. So the way I think of it is Müllerian, M for mother, which is a female, and those become the female ducts and glands. The Wolffian ducts are going to recede in females and therefore go away, but they develop in men, or the male infant, to become the male ducts and glands of the male reproductive system. Figure 17.2 shows us those 2 sets of ducts in the fetal development. So again, this is about 5 to 6 week old embryo. This is not after birth. So you have the Müllerian ducts here in purple, and the turquoise ducts there are the Wolffian ducts. So in the absence of a Y chromosome, which of course we know through inheritance, an X and an X inheritance is a female, and an X and a Y inheritance is a male. So in the absence of the Y chromosome, the Müllerian ducts grow and become the female reproductive system, and the Wolffian ducts are going to recede or go away and be resorbed. Now, in the presence of a Y chromosome, which means a male baby, the Wolffian ducts are going to grow and become the male reproductive structures, and the Müllerian ducts are going to recede and go away. All right, so from here we're going to focus on the female reproductive system and control of reproduction, and then at the end we will do the male reproductive structure. So again, the female genetically has inherited 2 X chromosomes, one from the mother and one from the father. And as we've just seen, that is going to determine which reproductive structures are going to develop. This is an external view of the female reproductive structure from figure 17.18. And we can see the opening of the vaginal canal or vagina. We also have the labia minora and the labia majora, the mons pubis, which was a fatty deposit over the pubic symphysis, and the clitoris, which is homologous to the penis in the male and is an erectile tissue which will engorge with blood during sexual excitement. Internal structures for the female reproductive system are going to include a gonad. And of course, a gonad from your general biology or human biology class was a structure that produces a sex cell. And of course, a sex cell has 1/2 of the chromosomes of a normal cell. And of course, if you put 1/2 of the chromosomes from the sex cell from the female and 1/2 of chromosomes from the sex cell of the male, we get one whole cell, which will then start dividing and dividing and dividing into trillions of cells big, and we have a new human. Okay. So the gonad will produce an egg, which is also known as an oocyte for the female, gamete. And of course, gamete is the scientific term for sex cell, half the chromosomes. Then we have the duct system, which begins with the fallopian tube, which of course you should know from anatomy is the common term. It is also known as oviduct or uterine tube. And that is connected to the modified duct known as the uterus, which can allow a fertilized egg to implant and foster that growing fetus for 9 months until birth. And then we have the vagina or vaginal canal, which leads to the external environment. From an anterior view, we can see that the ovary is going to release a gamete into the peritoneal cavity and is swept up into the oviduct. In anatomy, you should have learned that the fringed end of the oviduct is called fimbriae. They contain cells that have cilia on them that constantly create a microcurrent in the peritoneal fluid, which would sweep up an egg into the fallopian tube. The fallopian tube is attached to the uterus. The uterine cavity is where the growing embryo is going to be housed for 9 months. And then during labor or menses, if we don't have a fertilized egg, we use the passageway to the external environment known as the vagina. Now, the vagina has a natural and normal flora. Bacteria live in there and they are supposed to be there. We have a symbiotic relationship with those bacteria, meaning that we benefit from them and they benefit from us. The bacteria that form most of the normal flora of the vaginal canal is lactobacillus. And lactobacillus is going to inhabit our vaginal tissue and therefore outcompete pathogenic bacteria from the external environment from being able to take hold and proliferate within the tissue. And we feed lactobacillus as well as give it a house. The cervical tissue actually secretes glycogen. So yes, the cervix tissue secretes a sugar. And the lactobacillus can use that sugar and it turns it into lactic acid. which of course is part of anaerobic metabolism, and then the lactobacillus makes a little bit of ATP for itself. So we feed lactobacillus and they make lactic acid. Lactic acid is important to the health of the vaginal canal. Lactic acid is an acid, and therefore it causes the pH of the vaginal canal to be approximately pH 5 or so, and acidic conditions help to also decrease the proliferation of pathogenic bacteria. So we give them a house and food, they give us a pH condition that is not hospitable to most pathogenic organisms. Now that pH can actually change during the ovulation period of the ovarian cycle, which is around day 14, and we'll be discussing that in a little bit. So if the pH can increase, I think I might have said decrease. If I did, I apologize. Meaning that it can go to a higher number and become more alkaline, and in more alkaline conditions, the vaginal canal is more hospitable to the growth of other organisms, including yeast, which is a normal flora organism. It's just in very low quantities. Now, douching can be harmful to the vaginal canal because it is a flushing of the internal structure, and the flushing actually easily removes the lactobacillus, but organisms like yeast adhere to the tissue much more easily. And because of that, the yeast are left behind, and now most of the bacteria, which normally keep the yeast in check by outcompeting them for space in the tissue, are gone, and the yeast begin to proliferate, causing a yeast infection. Okay. Now the cervix of the uterus. The cervix means the neck of the uterus, and it is where the uterus is connected to the vaginal canal. And there are two parts to the cervix. There's a muscular part, which is the intermediate layer of the uterus. But then there is also a mucus or mucosal layer, and the mucosal cells are going to secrete what's called a mucus plug. That mucus plug is present throughout the menstrual cycle. Its thickness changes throughout the cycle. Okay. So the purpose of the plug, by the way, is not as a natural contraceptive. Sperm can get through the mucus plug. What it's actually there for is to keep organisms in the environment out of the uterus. We want the uterus to remain sterile because we have to grow a human in it. So the mucus plug is designed to keep bacteria that are housed in the vaginal canal and also open to the external environment, there are more bacteria, from being able to enter the uterus. Now, sperm are much bigger than bacteria, and sperm can get through the mucus plug. But the mucus plug will actually thin during ovulation, which just makes it easier for sperm to swim through, to be able to get past and enter the actual uterus, and it will thicken greatly during pregnancy. Because if we are housing a growing fetus, we definitely want to keep that internal environment sterile. Ferning is the process of the thinning of the mucus plug during ovulation, which makes it more easily passed by the sperm to enter the uterus. Human papillomavirus is a virus that can affect the reproductive system, and it can be detected by doing a pap smear. A pap smear is a test where a sample of cells from the cervix are taken and viewed under a microscope. And what the technician will be looking for are what are called neoplastic cells. So plastic means able to change and neo means new. So this means newly changed cells. So what they're doing is looking for cells that are not the normal shape of cervical cells. The problem with that is that the shape of cervical cells can change during the menstrual cycle. And at one point in the menstrual cycle, they can look like a cancerous cell shape. So it's always recommended not to do a pap smear the week before menstruation or during menstruation because the cells are usually a different shape. It's the couple of weeks prior to menstruation that is recommended to do a pap smear, and that is when the cells look most normal. Now, the uterus has 3 layers. The endometrium is the inside of the uterus, and it's therefore a mucosal lining. And of course, mucosa always lines open body cavities, and the reproductive system is open to the external environment. And endo means within. Metrium is referring to the uterus, so this is the layer within the uterus. That will be the layer that a fertilized egg or embryo will actually implant into and attach to the mother tissue. The myometrium is intermediate. So the myometrium means muscle of the uterus. This is smooth muscle, and that smooth muscle will perform peristaltic waves of varying degrees depending on the activity being performed. For example, during labor, it is very violent contractions to move the infant into the birth canal during labor. And during menstruation, they are more mild, although they may not always feel that way to women, to shed the inner lining of the uterus during menstruation. The outermost layer of the uterus is going to be perimetrium, which of course means surrounding the uterus, and it is the actual visceral peritoneum, which is the simple squamous lining of many of the organs in the peritoneal cavity. All right, from the uterus, sperm would enter the fallopian tubes or oviducts or uterine tubes, and those are attached to the uterus, and the other end of the oviduct is at the ovary. These tubes are approximately 4 inches in length and only about 1/4 of an inch in diameter, so they're not very big. And most of the cells lining the inside of the fallopian tube are simple columnar epithelia with cilia on them. And of course, you should have learned in your anatomy class that those cilia kind of act like a little conveyor belt, and they can roll back and then flick forward. And so during that, during that motion, anything that's in contact with the cilia, like an egg, would just get conveyor belted along those cilia to a destination, which of course would be the uterus. There are also other cells lining the inside of the oviduct that do not have cilia, and they secrete primarily types of fuel source, sugars, for the egg that is in the fallopian tube, because that egg is still a living cell and needs fuel to complete cellular respiration and make ATP. Now, the sperm are going to meet the egg in a region of the tube called the ampulla. And in anatomy, you should have learned that an ampulla is a widened end of a tube. And the ampulla of the oviduct or fallopian tube is at the ovary end of the duct. So if we're looking at the uterus and then the oviduct or fallopian tube, so here's the uterine cavity. And then this fallopian tube has its fringes, which we just discussed was called fimbriae. And that's right over the ovary, which is the gonad. So this wide end of the tube is known as the ampulla. Okay. And it's in the ampulla that an ovulated egg will sit, and a sperm that may be in the fallopian tube would normally fertilize the egg in that ampulla region. From there, the egg has to travel the length of the fallopian tube towards the uterus. It takes about 7 days to reach the uterus using the cilia action of the simple columnar cells, but also the fallopian tubes do have some smooth muscle in their walls and will do peristalsis towards the uterus as well to move the egg towards the uterus. Now, if the egg happens to implant in tubal tissue rather than in the endometrium of the uterus, where it should implant, it is known as an ectopic pregnancy, which means an implantation other than where it should happen. Tubal pregnancies can cause great inflammation or even damage to the fallopian tube and don't result in a successful pregnancy. Sometimes surgery is necessary to remove an ectopic pregnancy. Okay, so the egg will be ovulated from the ovary and swept up into the fallopian tube or oviduct. This region is the ampulla, where it's the wide end of the tube, whereas from anatomy you should have learned the narrow end attached to the uterus is called the isthmus. And if it is fertilized or not, it will still have to travel the length of the fallopian tube to the uterus. If it's not fertilized, of course, it will leave the body in menses. If it is, it will implant into the endometrium of the uterus. Okay, now, oogenesis. Oogenesis is the form of meiosis, which you should remember from your prerequisite general biology or human biology class, is a division process for the production of a gamete. And you are going to be responsible for meiosis for both a sperm and an egg in this class. So, the reason it's given its own term is because in meiosis, to produce an egg, you get one viable egg, whereas in meiosis for sperm, you get 4 viable sperm. So the terms oogenesis and spermatogenesis are the 2 different forms of meiosis, just indicating one is going to produce an egg and the other one will produce sperm. Now this figure is from your textbook, and it does give you a nice illustration of oogenesis. However, its description of chromosomes is not as elaborate as it should be to help students understand what's actually happening here. So we're going to do it together ourselves. Let's get a good old ice cream. All right, so oogenesis. Okay. So oogenesis is going to go through a couple of phases, but many years of the woman's life. The phases are going to be what happens during the fetal stages or as a fetus, childhood, and then the onset of menses or puberty. Okay. So we actually are going to develop all the eggs we're ever going to have in the ovary while we are a fetus. And what happens is we have stem cells with 46 chromosomes, the normal number of chromosomes for a human, and that stem cell can divide and give rise to a cell that is more specialized than it is. This stem cell is called an oogonium. That stem cell is going to go through mitosis. So when we do mitosis, we have to replicate our DNA, which gives us the original 46 plus their copies. So if you take 46 chromosomes and copy each one of them, effectively, how many strands of chromosomes do you have in there after that? Yeah, so 46 plus 46, or 46 times 2, would be 92 chromosomes. Now, you'll never see that in a textbook. So in a textbook, what you usually get is what we see in our book. So it's going to tell you that it's going to have 46, and then it's going to have to do mitosis here and produce more cells. But it's going to tell you that the original cell had 46 and its daughter cells had 46. Well, if you split a cell with 46 chromosomes, you don't get 46 after that. So you have to take into consideration that you're doing DNA replication here, which results in 46 chromosomes and their copies. So if you have 46 chromosomes and their copies, you get 92. When that splits, you get daughter cells that each have 46 chromosomes. So that cell is going to split in mitosis, and we get 2 cells each having 46 chromosomes. Okay. Now, the reason we have to do mitosis first is because if we didn't, if that oogonium divided and produced 2 cells that would continue through meiosis, you'd run out of oogonium. So what happens is the oogonium replicates its DNA and completes mitosis. One of those will continue as an oogonium and keep doing that. Oogonium. The other one is called a primary oocyte. So it is more specialized than the oogonium, and it is now dedicated to becoming an egg. So this one will keep doing this process and making more primary oocytes and more oogonium while we're a fetus, and the other daughter cell will continue through the process. Now, those primary oocytes are going to be arrested. What does arrested mean? Stopped in meiosis 1. Okay, and I'm going to have to move this line down just a little bit so that I can write something else there. Okay, so I'm putting the same stuff back up there. I'm just moving it down. Okay, so that primary oocyte is going to start meiosis, and of course meiosis begins with replicating our DNA, but then it will go through 2 divisions, and so if 1 division results in cells with 46 chromosomes, how many will 2 divisions result in? Yeah, 23, which is the amount a gamete has. So to start meiosis, it's going to replicate its DNA, which will give us 46 chromosomes and their copies. Okay, but then it's going to sit there all the way through childhood. Meiosis 1, that first division, cannot finish unless the egg is ovulated from the ovary. So it sits there for a little over a decade until the onset of menses during puberty. So here's our primary oocyte with 92 strands of chromosomes, the 46 original and then their replications. And once a month, about 10 of these oocytes will be stimulated to continue into meiosis. So ovulation is what causes the first division of meiosis to finish. So we're going to have completion of meiosis 1. So phase 1 of meiosis, and that happens when the ovary ovulates that oocyte. Ovulation. That triggers the meiosis stages to finish, so prophase, metaphase, anaphase, telophase, and you end up with 2 daughter cells. But they aren't going to split equally. One of those cells is going to get basically all of the cytoplasm and the organelles. The other one is going to have minimum cytoplasm and chromosomes. So this one is called a polar body. It still has 23 chromosomes. The chromosomes do split like it's supposed to. They line up on the metaphase plate and then they get pulled to the poles by the mitotic spindle during anaphase, and so on and so forth. There's still 23 chromosomes in there, but you don't have the rest of the cellular machinery to go on after that. That polar body cannot be fertilized. The other daughter cell has all the cytoplasm and the organelles and 46 chromosomes. That one is now called the secondary oocyte. Now, that secondary oocyte is going to be arrested in meiosis II. So in the second division phase of meiosis. It can only finish that division phase if it's fertilized. So it gets ovulated, and now it's in the ampulla of the fallopian tube. If a sperm happens to fertilize the egg, then it will finish its division. You'll end up with one egg that has 23 chromosomes of its own, plus the 23 chromosomes that the sperm delivered to it, and a second polar body that has 23 chromosomes. Remember, 46 divided by 2 is 23, so each daughter cell will have 23, but one of them will also have 23 from the sperm. What the heck? Stop it. So this guy still has 23 chromosomes. Okay, but it can't be fertilized. It's just like getting rid of the waste so that the rest of it can go on and start dividing and become a morula and a blastula and an embryo and fetal stages and blah, blah, blah until birth. Okay, so that is another polar body, and it can't be fertilized. Okay, so the other daughter cell has its 23 chromosomes plus the 23 from the sperm, and it's going to divide and divide and divide, and we have a human. Why is that happening? All right, now, a couple more things about this process. That first polar body may split in half. If that happens, oh shoot, I made an error back here. This first polar body has half of 92, which is 46, not 23, Mrs. Gibbs. That polar body may divide, in which case you'd get two polar bodies, each with 23 chromosomes. So through oogenesis, you can have 2 or 3 polar bodies, because that first polar body splitting may happen or it may not. Doesn't matter if it does, polar bodies can't be fertilized, it just might happen. So with oogenesis, you get 2 to 3 polar bodies. 1 fertile egg. Okay, stop it. All right, now, what happens if that secondary oocyte doesn't get fertilized? If it doesn't get fertilized, it doesn't finish this division. It will actually leave the body in menses stuck in meiosis II. It will leave the body with 46 chromosomes in the middle of the second division phase of meiosis and will never complete its division. Okay, so if no fertilization, the secondary oocyte leaves the body in menses, arrested in meiosis II. Alrighty, so there you go. You won't be asked to replicate this process on an essay. You will be expected to be able to answer questions as to what happens. So for example, I can ask you what must happen for a primary oocyte to complete meiosis 1, and you would have to know that ovulation has to happen. I could ask you how many polar bodies results from oogenesis. I could ask you what must happen for a secondary oocyte to complete meiosis 2. I could ask you how many chromosomes are in an arrested primary oocyte in the fetus, and so on and so forth. So you are expected to understand the information that's been given here. You will not be asked to diagram this out. Okay. Now, those oocytes grow inside of a cellular structure called a follicle. And the follicle is going to start out as a fetal stage as simple squamous cells surrounding an oocyte. Then, once a month, about 10 of those will be stimulated to develop. And the simple squamous cells are going to become cuboidal. So they aren't making more cells. We're not exchanging these cells. The simple squamous cells will stop using the set of genes to be squamous and start using a set of genes to be cuboidal. When that happens, these cells are now called follicle cells or follicular cells. Okay. Those follicular cells are going to begin dividing and doing mitosis. The daughter cells of the follicle cells are called granulosa. So now you'll have the oocyte, and the granulosa will start being produced between the follicle cell that's doing mitosis and the oocyte. So as those follicle cells are doing mitosis, you'll get layers of granulosa between the oocyte and the cells that are doing mitosis, the follicle cells. So the follicle cells will end up on the outside. Okay. So those are still the follicle cells. These ones are going to be the granulosa. Now, the granulosa cells are going to do two things. The granulosa are going to secrete an enzyme called aromatase. They will also secrete fluid and develop the antrum. So you should have learned antrum in your anatomy class. It is the fluid that builds up around the egg in a developing follicle. When that fluid builds up so much that the tissue surrounding it can no longer hold it back, it ruptures. And that is actually ovulation, is the rupturing of the follicle that can no longer hold back this antrum, and the egg on the inside is released from the surface of the ovary. Now, surrounding that follicle are going to be additional cells called fecal cells. So we're going to draw this out a little bit bigger and show you what the relationship between the theca and the granulosa cells are. So this is going to be our developing oocyte. Okay. And then, of course, surrounding that oocyte, we have layers of granulosa that were produced by the original follicular cells doing mitosis. Okay, so these are all granulosa. And surrounding them are the original cells that did mitosis to produce the granulosa. So here are your original simple cuboidal cells called the follicular cells that did mitosis. Okay, you get the idea. Okay. Now, in the tissue surrounding the follicle in the ovary, you have cells that surround the follicle in the extracellular tissue. These cells are called theca, or thecal cells. Okay. Now, the granulosa, as we described, produces an enzyme called aromatase. I want to make that a little bit shorter. And the theca actually produce androgen. So androgen is a class of chemicals. Okay. And the androgen that is secreted is testosterone. All right, so yes, a female cell secretes testosterone. That testosterone is worked on by the enzyme aromatase to make estrogen. So that's how the female gonad makes estrogen. The granulosa make an enzyme that turn the testosterone from the theca cells into estrogen. It's called aromatase because it cleaves aromatic rings in the cholesterol-based testosterone. You won't be asked that on test, but I'm just explaining where the name aromatase comes from. All right, and then once you have estrogen, that estrogen is used to control the growth of the follicle and also what will happen in the uterus during this time. Okay, so the onset of menarche. So menarche means the beginning of puberty and the cyclic process that the ovary will go through to develop an oocyte that will be ovulated once a month. Up until puberty, gonadotropin releasing hormone is inhibited by estrogen. And you might be asking, well, where is the estrogen coming from if we haven't turned on the ovary yet? Well, there's other sources of estrogen in the human body, one of them being the adrenal gland. And so the small amount of estrogen is going to inhibit the hypothalamus. So the hypothalamus won't release gonadotropin releasing hormone. And you should know from our endocrine lectures that gonadotropin releasing hormone goes to the anterior pituitary to cause the release of FSH and LH, which goes to the gonad. So if this is inhibited, then none of this is happening until puberty. Okay? But at puberty, the hypothalamus becomes less sensitive to estrogen. And if it's less sensitive to estrogen, it's not inhibited. Okay? So if estrogen has to bind to a receptor intracellularly, we know that estrogen is a steroid hormone, so it will follow the direct gene pathway. For a cell to be less sensitive to a hormone's action, what must it do? Make less receptor for it. So the hypothalamus cells start making less receptors, and therefore estrogen can't have much of an effect on the hypothalamus. And boom, it starts releasing gonadotropin releasing hormone, which will stimulate the anterior pituitary. So we're going to do what happens in the ovary first, and then we'll follow that with what happens in the uterus during a 28-day cycle. So for the ovarian phases, the things that happen in the ovary, which of course has to do with the follicle leading to ovulation, and then implantation if fertilized. Okay, so this is ovary stuff. Later we'll do uterine stuff. Okay, so the ovarian phases are divided into two-week phases. Days 1 through 14 are called the follicular phase. This is when the follicle develops and leads to ovulation. Okay, so this is during the time where your follicular cells are doing mitosis and making granulosa. The granulosa are making aromatase and also building an antrum, which makes the follicle bigger and bigger and bigger until it can no longer hold back the fluid of the antrum and it ruptures, releasing the egg into the peritoneal space. After that, the follicle is going to start using a different set of genes. So those granulosa and follicle cells will start using a different set of genes from their DNA and take on a new role, and that will be during the luteal phase. So phases 14 to 28 is the luteal phase, where the follicle becomes the corpus luteum and secretes progesterone and will still secrete estrogen. And that's to control the ovary during the period of time that we're waiting to see if an egg will be fertilized, and also to control the uterus and keep it from contracting until a fertilized egg has an opportunity to implant into the endometrium. And then this cycle repeats every 28 days. Now, of course, that's the average. There is a range. Some women have shorter cycles, some women have longer cycles, some women have erratic cycles, etc. The average is about 28 days. And as I mentioned, for each menstrual cycle, about 10 follicles will mature. But only one egg normally is ovulated. What happens is the first egg to be ovulated causes a chemical reaction in the ovary where the other 9 follicles are resorbed, just taken back up by the ovary and disintegrated. So we typically have one ovulation. All right, oops, we're going to do this ourselves. So we're going to be building onto this chart as we go through the lecture. And above this line is going to be ovarian phases. And below the line, we'll add uterine phases later. Okay, so we're going to divide the 28-day cycle into 4 weeks, and a week is 7 days. So this would be day 0 of the cycle, and then day 7, day 14, day 21, and then day 28. Now, ovulation happens at day 14 for the average cycle. And the follicle develops up until ovulation, so days 0 to 14. That is our follicular phase. That is happening inside the ovary. After ovulation, that follicle becomes the luteal body, or the corpus luteum. So it will go through the luteal phase. Okay. All right. So, yeah. So what we're seeing here is the first 2 weeks are the follicular phase of the ovary, and the second 2 weeks are the luteal phase of the ovary. I just realized that they moved into the second cycle there. So the first 2 weeks are the follicular phase, the second two, oh no, okay, just kidding. The second 2 weeks are the luteal phase. So we're going to describe what's going to happen in this phase and this phase of the ovary, and then we'll discuss the uterus. So in the follicular phase, we're going to have gonadotropin-releasing hormone act on the anterior pituitary. And that's going to cause the anterior pituitary to release follicle-stimulating hormone. That is going to have an effect on the granulosa, and luteinizing hormone, which has an effect on the theca. Now, when luteinizing hormone binds to the thecal cells, luteinizing hormone is a protein hormone, so it will bind to extracellular receptors, and it will activate a G protein pathway, which results in protein kinase enzymes that result in phosphorylation. In this case, it's phosphorylation of the phosphorylation of transporters that uptake cholesterol into the cell. And the job of thecal cells is to produce androgen, specifically testosterone. And so we need a lot of cholesterol to produce that testosterone. Then enzymes inside of the thecal cell will make will turn cholesterol into testosterone and secrete testosterone. The aromatase from the granulosa cells will then turn the testosterone into estrogen, as we discussed. So, I've got to figure out where I am here. Here's what we're going to do. This is going to be the hormonal control of the follicular phase of the ovary. Okay, so here we go. We start with the hypothalamus. So the hypothalamus is going to secrete gonadotropin-releasing hormone. Gonadotropin-releasing hormone will travel by blood and act on the anterior pituitary. Stop it. Pituitary. The anterior pituitary will then release follicle-stimulating hormone and luteinizing hormone into the blood. Follicle-stimulating hormone is going to act on the granulosa cells. And it's going to cause the granulosa to do two things. One is to secrete aromatase, the enzyme that will turn testosterone into estrogen. Aromatase. The other is to secrete inhibin. If this molecule is called inhibin, what do you think it will do? Inhibit. That hormone enters the bloodstream and turns off the anterior pituitary. Once we get follicle-stimulating hormone and luteinizing hormone, the product of that, inhibin from the granulosa, is going to negatively feed back and turn off the anterior pituitary. Now, luteinizing hormone acts on the theca. What did the theca produce? Testosterone. And testosterone is going to be turned into estrogen by aromatase. So now we have the presence of estrogen, and this is going to be in small amounts because the follicle is still small at this point. Small amounts from a small follicle. Okay. Estrogen is also going to be a negative feedback, just like we talked about with the endocrine system. So estrogen will enter the blood supply, and it will have a negative feedback at the anterior pituitary and the hypothalamus. So now we're also stopping gonadotropin-releasing hormone from even stimulating the anterior pituitary. But as the days go on through the follicular phase, we get follicle growth. So if we get a bigger and bigger follicle, we get more and more estrogen. So now we have a large amount of estrogen. And interestingly enough, a large amount of estrogen is actually a positive feedback to the hypothalamus. So where a small amount of estrogen present in the bloodstream turns off the hypothalamus, a large amount will turn it on. I shouldn't say turn off the hypothalamus. It won't turn off all the function of the hypothalamus. It'll just stop it from secreting gonadotropin-releasing hormone. But a large amount of estrogen will stimulate the hypothalamus. So the second round of hypothalamus, when it's stimulated by this estrogen coming from a large follicle, is to release gonadotropin-releasing hormone again. That's going to then cause the anterior pituitary to release FSH and LH again. You'll get a fair amount of FSH, which is called the FSH spike, but you'll get a huge amount of LH, which is called the LH surge. The FSH spike and the LH surge cause such a large amount of antrum to be secreted by the granulosa that the follicle can no longer hold back the pressure of the antrum and it ruptures, which is what we call ovulation. Okay, so this is a primordial follicle. That is what is developed at birth, I should say before birth when we're a fetus, and it sits that way all the way until puberty. At puberty, approximately 10 of these follicles are stimulated to develop. The squamous cells will become cuboidal, and they will start to do mitosis and produce the granulosa cells. The granulosa cells are going to start secreting, and that secretion builds up around the egg and it is called the antrum. As the follicle grows and we get more and more granulosa, we get more and more secretion and a giant antrum. Eventually, the follicle can't hold back that fluid and it will rupture, and the egg inside of the follicle will be ovulated. Now, figure 17.23 shows us that luteinizing hormone acts on the theca, which synthesizes androgens, and the follicle stimulating hormone acts on the granulosa and causes it to produce aromatase. They don't go into the detail, they just say convert androgen. Well, it does that with aromatase, and we get estrogen. So the estrogen in the plasma from a small follicle, it provides a negative feedback to the anterior pituitary. This should say and the hypothalamus. Okay. Inhibin is going to be a negative feedback to the anterior pituitary only and inhibits the release of follicle stimulating hormone. But then as that follicle grows, we get a lot of estrogen, which causes the FSH spike and the LH surge. And it's thought that it's really more the LH surge that makes a difference. It's going to cause the granulosa to secrete a lot of antrum. That is going to rupture the follicle, cause the completion of the first stage of meiosis, and the egg exits the follicle, which is what we call ovulation, and that typically happens at around day 14. The follicle will then become what's called the corpus luteum, so the luteinizing hormone causes that. And then additional luteinizing hormone and follicle stimulating hormone secretion is prevented by declining gonadotropin levels. So basically, once the follicle becomes a corpus luteum, we're not secreting large amounts of estrogen. It's secreting small amounts of estrogen, which will now inhibit the hypothalamus again, and it will stop producing gonadotropin releasing hormone, which will stop stimulating the pituitary from releasing follicle stimulating hormone and luteinizing hormone. So this figure is very similar to what we drew out together. So this is the hypothalamus secreting gonadotropin releasing hormone. That is going to cause the anterior pituitary to secrete FSH and LH. LH causes the secretion of testosterone from the theca, and FSH causes the secretion of aromatase from the granulosa. The testosterone is turned into estrogen by aromatase. The granulosa will also secrete inhibin, which will be a negative feedback to the anterior pituitary. But as we increase the amount of estrogen, it is a that's on the next slide. The small amount of estrogen that is currently now in the follicle is also a negative feedback to the pituitary and to the hypothalamus. But then this follicle is going to grow, and as it gets bigger, we get more and more estrogen. So as we get large amounts of estrogen, that's going to be positive feedback to the pituitary and the hypothalamus. We get a new secretion of gonadotropin releasing hormone, which causes the FSH spike, which is not thought of as important as the luteinizing hormone surge, LH surge. That LH surge causes the ovulation. After ovulation, the follicle becomes the corpus luteum. The corpus luteum is going to secrete both progesterone and estrogen. So the corpus luteum is going to begin the luteal phase, which is days 14 to 28. Progesterone is often written as P4. Will now be secreted from the corpus luteum in addition to estrogen and inhibin. So all three of these, estrogen in low amounts, inhibin, and progesterone, will provide various negative feedbacks to the hypothalamus and the pituitary. That is what prevents any more development of oocytes during this time. So basically the hypothalamus and the pituitary are shut down for about a week. That way, if we have a fertilized egg implant into the uterus, we don't have new ovulations that are going to produce compounding pregnancies. Now, after that 7 days, the corpus luteum begins to die. And if the corpus luteum dies, there's no more estrogen, progesterone, or inhibin. And if there's no more of those, the hypothalamus is like, woohoo, I get to do my thing. Thalamus. And we get more gonadotropin-releasing hormone, and that whole cycle starts all over again. We're going to describe that in more detail in a moment. Now, what is progesterone doing while it is being secreted from the corpus luteum? The progesterone is a negative feedback to the hypothalamus and stops gonadotropin-releasing hormone, but it also travels by blood to the uterus, and it does what's called quieting the myometrium, meaning that it is inhibiting contractions. And it will do that as long as progesterone is in the bloodstream. It will be in the bloodstream as long as the corpus luteum is alive. This gives a long enough period of time for an egg to implant and begin secreting hormone that will tell the uterus itself to continue to be quiet. If there's no implanted fertilized egg, the corpus luteum dies, and now there's no inhibition to the myometrium of the uterus, and the contractions of menses will start. Okay. In addition, progesterone will also act on the mammary tissue, along with estrogen, and it will stimulate the production of mammary tissue. Now, PMS, or a condition that is premenstrual syndrome, is thought of to be as a result of erratic progesterone levels, and erratic progesterone levels can result in early or late contractions or the onset of menses, and also is thought to cause other symptoms such as bloating and so forth, leading up to the menstruation. Okay. This figure puts the whole thing together as well, including the corpus luteum during the luteal phase. So the corpus luteum is secreting progesterone and estrogen and inhibin, and therefore the hypothalamus and the anterior pituitary are shut down, giving a potentially fertilized egg an opportunity to implant before menses would begin and stopping additional development of follicles during that time. Now, if a fertilized egg implants, we get secretion of human chorionic gonadotropin. The little h does mean human. And the reason that it has a little h in front of it is because chorionic gonadotropin wasn't originally discovered in humans. So this is designated that it's coming from human physiology. So that ball of cells that is dividing will secrete human chorionic gonadotropin. That keeps the corpus luteum from dying. If the corpus luteum doesn't die, we keep getting progesterone for 9 months. Not only for 9 months is the uterus quiet so that we can grow an infant, but we continue to inhibit FSH and LH production, and therefore no follicle activity. So we get one baby growing at a time. Again, it takes about 7 days to reach the uterine lining for implantation, and it will implant into the endometrium layer. All right, now the uterus, so the endometrial cycle. So this isn't the ovary, this is what's happening in the uterus tissue now. So days 7 through, so we're going to start with day 7. Days 1 through 7 are menses. So we're going to go back and add these to our diagram back here. Ooh, way back here. Way back here. Okay. So in the uterine phases, days 1 through 7 are the bleeding phase. So that's menses. Once we start getting estrogen production from newly developing follicles, the uterus will begin to grow its inner lining. That is known as the proliferative phase. So to proliferate means to grow. So the thickness of the inside of the uterus is growing at that time. Then at day 14, we have ovulation. And following ovulation is the secretion of progesterone. Days 14 to 28 are known as the secretory phase because we are secreting substances from the uterine lining that will support a growing ball of cells until a placenta forms and the growing embryo can receive nourishment from the blood supply of the mother. Okay, so those are the endometrial or uterine phases. We're going to start here at day 7 with proliferative, then we'll go through secretory, and then end at menses at the beginning of the cycle. Okay. So days 7 to 14 are the proliferative phases. During this time, estrogen is being produced by a follicle from the ovary. And that estrogen enters the blood supply and will influence arteries called spiral arteries because they're coiled to grow through the endometrium layer, which is something that doesn't always happen because remember, endometrium is epithelia, and you should have learned from your general class that epithelia is avascular. But because we're going to have an implantation of an embryo, we need a blood supply there to provide that growing ball of cells with what it needs. The estrogen will also act as a transcription factor to bind to the DNA and cause the transcription and translation of receptors for progesterone so that the receptors are there by the time we start getting progesterone at day 14. Now, days 14 to 28 are the secretory phase. During the secretory phase, we start getting progesterone, and progesterone continues to influence the growth of spiral arteries. We want a nice rich blood supply in anticipation of an implantation of an embryo. Also, the mucosa or endometrium of the uterus will begin secreting substances to support a growing ball of cells, such as glycogen, and this is why it's called the secretory phase. During this time, the progesterone also causes the cervical cells to produce a thicker mucus, therefore thickening the mucus plug, preventing bacteria from getting into the uterus, where we may have an implanting embryo. Next. Okay, now, halfway through the secretory phase, the corpus luteum dies. And becomes a scar on the ovary called the corpus albicans, which means the white body, which is a scar. That will happen if there's no implanted embryo secreting human chorionic gonadotropin. So if that happens, the corpus luteum dies, which means we no longer have progesterone supporting the tissue of the uterus, and the myometrium is no longer quiet, and we will slough the functional layer of the endometrium. Then by day 1, the sloughing has occurred to an extent that the spiral arteries are exposed, and that's where the bleeding from menstruation comes from. And it takes approximately 7 days of menstruation for mitosis of the endometrium to heal and cover those arteries where the bleeding stops, and then we enter into the proliferative phase again at day 8. Okay, so we want to go back to our chart. Sorry. Okay, so the thickness of the uterus during the uterine cycles would look something like this. It's going to be low during menses because we sloughed it. Then it's going to grow during the proliferative phase when estrogen is encouraging the growth of the endometrium. Then it's going to remain high while the corpus luteum is alive. But then the corpus luteum begins to die about halfway through the secretory phase, and we begin the sloughing and end up at the same level that we would start at the beginning of menses again. So this is uterine thickness throughout the uterine phases. Okay, now that thickness is because of the presence of estrogen and progesterone. The presence of estrogen and progesterone are due to the presence of follicle-stimulating hormone, luteinizing hormone, and gonadotropin-releasing hormone. So we are going to add the levels of those hormones to this diagram in the ovarian phases. So during the follicular phase, we have gonadotropin-releasing hormone, and then it's going to start to become inhibited for a little bit while that follicle is small. Then the follicle the follicle gets big and we have a spike, and then it's going to drop again and be low because it's inhibited during the luteal phase. Okay, so this was gonadotropin releasing hormone. Now, whatever gonadotropin releasing hormone does, follicle stimulating hormone and luteinizing hormone are going to follow because gonadotropin releasing hormone stimulates the pituitary to release those two hormones. So luteinizing hormone and follicle stimulating hormone are going to follow that pathway of gonadotropin releasing hormone just a little bit behind it because the gonadotropin releasing hormone stimulates the pituitary. So luteinizing hormone and follicle stimulating hormone is going to come a little bit after. And then we're going to get our spike, and then it's going to decrease back to low levels for the remainder of the luteal phase. Then we also have estrogen. Estrogen is basically going to follow the same pattern as luteinizing hormone and follicle stimulating hormone, because luteinizing hormone and follicle stimulating hormone cause the release of testosterone and aromatase, which makes estrogen. So estrogen is going to start to build and build, and then it's going to spike and then be low during the beginning of the luteal phase. And once that corpus luteum is in high gear, it's going to rise a little bit again and then fall when the corpus luteum dies. And we have progesterone. Progesterone is basically none during the follicle phase because the follicle doesn't secrete progesterone. The luteal body does. So once we start getting a luteal body, that's going to rise, maintain for a while, and then fall when the corpus luteum dies. So those traces just show you where those hormones are rising and falling throughout the ovarian cycle. When estrogen is high, we are stimulating the growth of the uterus. When progesterone is high, we're maintaining the thickness of the uterus. When estrogen and progesterone are low, the thickness of the uterus is going to drop. Okay. Okay, let's get back to where we were. Okay, so a nice illustration of what's happening is also provided to you in your textbook in figure 17.27. So you have your ovarian phases. So we have a growing follicle throughout days 1 through 14. At day 14, we have ovulation. After ovulation, the follicle becomes the corpus luteum. Corpus luteum will secrete estrogen, progesterone, and inhibin, which is not on this diagram. But then about halfway through the secretory phase, it's going to die and become the corpus albicans. When that dies, we're no longer supporting the uterine lining, and it's going to drop off and slough during menses. Okay. When estrogen is being produced, the uterus is being stimulated to grow. And when progesterone and estrogen are present, we maintain the uterus. Figure 17.22 also shows you the traces of the hormones that we discussed. FSH and LH are basically going to do the same thing because they are going to be stimulated by gonadotropin-releasing hormone. So they'll rise and then fall a little bit, and then they'll peak, which is the FSH surge, and the FSH spike, LH surge, which causes ovulation at day 14. Okay, and then those are going to fall when the corpus luteum is present and progesterone and estrogen and inhibin are inhibiting the hypothalamus and the pituitary. Okay, estrogen is going to build and build and build, spike, and then drop. And then when we get a good corpus luteum, it's going to be secreted again. Corpus luteum dies and it drops. Progesterone is basically none until we get ovulation and the corpus luteum develops progesterone and it's high, and then corpus luteum dies and it's going to drop. Now menopause. Menopause is defined as having no menstruation at all for a year for one whole year, okay, 12 months in a row. And so there will be a period of time where menstruation becomes less frequent. It may only occur 8 times that year or 4 times that year, but when it's finally none for a 1-year period of time, consistent, it is officially menopause. This is happening because the ovaries are becoming less responsive to gonadotropin-releasing hormone, which means we are reducing receptor production. Basically, the ovaries stop using their genes for receptors for gonadotropin-releasing hormone. If you don't have a receptor for it, the hormone can't have an action there. And if we don't have ovary function, we don't make estrogen. And if we don't make estrogen, we are no longer stimulating follicle growth. The theca are no longer producing testosterone, and the ovulations and the menses are going to stop. That's where we're going to stop this lecture. We'll have the male reproductive system lecture as a separate lecture.